Welcome everyone. My name is Irene Suarez-Martinez, and I am a researcher at Curtin University in Australia. I would like to thank Curtin University and the Australian Carbon Society for supporting this webinar. And I welcome you all and thank you for making the time today to join us. Just a reminder that this seminar is being recorded and will be uploaded in YouTube, so please stay muted while the talks are happening. Today we have another round of 4 presenters, those short award talks by early career researchers. We are going to have Dr. Huseyin Karahan, which is the winner of the Carbon Journal Thesis Award in 2019. We're also going to have Dr. Javier Quilez-Bermejo, who won the Carbon Journal Thesis Award in 2021. Also, Dr. Jacob Martin, who also won the winner of the Carbon Journal Thesis Award in 2021. And the last talk will be by Dr. Agnieszka Chojnacka, which is the winner of the Polish Chemical Society Presentation Award. So I think we all know how this runs now, but a quick reminder. Please stay muted while the talks are going on. Later on, we will ask all the questions at the very end of the 4 talks. So you can post a question on the chat, and then you can raise your hand if you prefer to talk and unmute yourself at that time when I mention your name. At the end of, once the questions are all done, we will all gather in Gather Town. With the link will be provided in the chat. So without further ado, I don't think there is anything else to say. We will just commence the first talk by Dr. Huseyin Karahan.
Hello everyone, I am Huseyin Karahan. Today I will tell you about my research on antimicrobial graphene materials in the last couple of years. But let me first introduce myself a little bit. I'm originally from Turkey. I earned my bachelor's and master's degrees in Istanbul. Then I moved to Singapore. There I worked on antimicrobial activity of graphenes during my doctoral studies. Later I shifted my focus to membrane technologies, and I also visited Sydney for research on both topics. I spent my COVID days in Kyoto working on carbon-based membranes primarily. In this talk, I will focus on my PhD and early postdoc activities mainly. Our initial motivation in investigating the antimicrobial activity of graphene oxide was to help get prepared for a possible future pandemic of bacterial origin. I used to explain to people the devastating impacts of pandemics, which is no longer news to anyone, unfortunately. But the difference of the expected bacterial pandemic is that it will likely be because of a multi-drug resistant bacterial species, and we can do something about it now by reducing our antibiotics abuse and developing alternative antimicrobial agents. As antibiotic substitutes for certain applications, nanosilver-based products were already in the market when I started this line of research, and nanocarbons, including graphenes, were emerging as antimicrobial agents as well. So I was wondering if graphene materials will join nanosilver one day in the future. For this to happen, we surely need to understand graphene-bacteria interactions much better. The impact of materials properties, such as layer number, lateral dimension, and oxidation level, was studied much more extensively. We could even build some structure property activity relationships later on. But there are 2 more less studied players in these interactions, cells and the environment, so I focused on these primarily. I started by thinking about why graphene materials often show lower antibacterial activity against gram-negative bacteria. Here you see a general overview of a gram-positive and a gram-negative bacterial cell. Basically, gram-positive cells have 2 layers in their cellular envelope, a thick peptidoglycan layer and an inner membrane. On the other hand, gram-negative cells have three layers: an outer membrane, a thin peptidoglycan layer, and an inner membrane. We thought that this outer membrane might be the key as an additional armor and decided to attack it. And we disturbed the outer membranes of gram-negative bacteria using an old trick, water shock, also known as hypoosmotic shock. On the left, you see happy bacterial cells exposed to salty water that they like. The salt concentration inside and outside of the cells are close. As a result, cell surfaces are intact and smooth. When the salt concentration is reduced, however, some cells burst, and intact cells are also damaged sublethally. Based on this principle, I hypothesized that we could observe higher antibacterial activity if we treat bacterial cells with graphene oxide and water shock at the same time. And it worked as expected. In this graph on the left, you see the survival rate of bacterial cells as a function of the environmental salt concentration. Under the same graphene oxide concentration, as we reduce the salt concentration, the antibacterial activity increased markedly. Microscopy studies also provided similar observations and confirmed our hypothesis. So, graphene oxide shows higher antibacterial activity on envelope damaged cells by water shock effect. Then, to increase the salt stability of graphene oxide, we combined it with a well-known polymeric stabilizer, a three-block copolymer of polyethylene oxide and polypropylene oxide, better known as pluronics. When there is no stabilizer, graphene oxide aggregates in time in the presence of cations, but in contrast, pluronic stabilized graphene oxide shows much higher colloidal stability when exposed to salt. As you see here, particle size distribution remains unchanged in time. But we also observed something a bit unexpected. As we increase the pluronic concentration, the antibacterial activity of graphene oxide boosted. This exciting observation is likely because of the combination of the increased colloidal stability of graphene oxide and the osmotic pressure of polymers on the cells. And very interestingly, the mixture of pluronic and graphene oxide showed lower toxicity to human skin cells. Under the experimental conditions we studied, graphene oxide killed around 12% of the cells, while the combination of pluronic and graphene oxide killed less than 6%. This observation is a bit puzzling, but mammalian cells are known to be jello-like substances, and they have extra support of a microtubular cytoskeleton, so we think that they might be handling the osmotic pressure effects a bit better. Finding such a simple, effective, and potentially biocompatible formulation was exciting for us, but unfortunately, pluronic stabilized graphene oxide showed less activity on gram-positive cells. As I emphasized earlier, gram-positive cells do not have outer membranes and have much thicker peptidoglycan layers. Therefore, we wanted to explore the role of peptidoglycan layer further. We exploited another core microbiological concept and modulated the physiological conditions of our cells. In a nutshell, we grew our cells more than usual and they built thicker peptidoglycan layers. You may consider this approach as a way of comparing young and adult cells. We usually test only young cells in standard antimicrobial tests. The results were so revealing. Very obviously, young cells die away even at low graphene oxide concentrations. In contrast, mature cells are much more resistant to graphene oxide, and we were able to verify this conclusion with different complementary techniques. So the question was, how to kill those mature cells? Naturally, we tried pluronic graphene oxide mixtures first. It helped a bit on some species, but basically failed. Bacterial cells generally exhibit mature physiological traits in natural environments, so we have some ongoing works to attack those type of cells and already have some promising results. In the near future, we hope to unlock real-life applications such as surface disinfection and even biomedical applications such as wound care. In fact, there are many more potential biomedical applications of graphene materials, which we have reviewed in this progress report. In addition to biomedical applications, the antimicrobial activity of graphene materials and their composites is also interesting for environmental applications. In a recent study, for example, we demonstrated that graphene oxide-based sponges can function as a point-of-use filter. And when it is functionalized with a specially designed quaternary ammonium compound, the resulting assemblies showed very promising antibacterial activity against gram-negative pathogens. For more information on nanocarbon-based water disinfection applications, I will recommend another review we contributed. Besides, if you are interested in membrane technologies especially, you might find our review on MXene-based membranes useful, which also covers antimicrobial applications of MXene materials, which can be considered as a cousin of graphene materials. I would like to thank all my mentors, mentees, and colleagues with a special emphasis on UNCHEN. There are several organizations to mention, but the support of A-STAR under the SINGA program was particularly critical for these studies. And I'm also grateful to JSPS for the JSPS Fellowship. Lastly, I would like to thank the organizers for their kind invitation, and thank you very much for your time and interest.
Hello to everyone. My name is Javier Quilez-Bermejo. I'm a postdoctoral researcher at the Institut Jean Lamour in the University of Leuven, and today I will show you a small part of my work that is entitled Easy Enrichment of Graphitic Nitrogen to Prepare Highly Catalytic Carbons for the Oxygen Reduction Reaction. Before starting the presentation, I'd like to thank the organization, especially to Jacob Martin, for the opportunity and for the invitation to be here today giving this talk. As you all probably know, fossil fuels play a dominant role in the energy supply, reaching almost 90% of the global energy demand. However, they present some important limitations, such as all the environmental issues that are related to their use. In this sense, fuel cells arise as a promising replacement for the current combustion engine because of their high efficiency towards energy production and low polluting emissions. As we can see in this slide, a fuel cell is an electrochemical device that converts chemical energy into electricity. However, the presence of electrocatalysis is necessary with the aim of increasing the kinetics of the adjoint oxidation reaction in the anode electrode and oxygen reduction reaction in the cathode electrode. One of the main limitations of this device is the use of platinum as electrocatalyst for both anode and cathode reactions, since, as we can see in the next illustration, the platinum needed to replace all the combustion engine by fuel cells, it's much bigger than the platinum we can find in nature. And this is especially remarkable in the cathode electrode where the oxygen reduction reaction takes place, since the 90% of the total platinum content is located. This is why most of the scientists who work on this matter try to focus their research on the development of new materials that are capable of reducing or replacing the platinum content in the cathode electrode of the fuel cells, where the oxygen reduction reaction occurs. All these alternatives can be classified into two different groups. First, the metal-containing catalyst, and the second, and to my mind the most innovative alternative, the use of metal-free catalysts, especially those that are based on carbon materials. Because these kind of electrodes are low-cost electrodes with high stability and a strong resistance towards poisoning with methanol or carbon monoxide. The main problem of this carbon material is the low catalytic activity towards the oxygen reduction reaction. In order to increase this catalytic activity, the introduction of heteroatoms or even defects are mandatory with the aim of disturbing the electronic density of the carbon material, leading to the chemisation and reduction of the oxygen molecules. This is especially important by using nitrogen-doped carbon material, which have proven to be capable of reducing very efficiently the oxygen molecules. But in experimental chemistry, when we prepare a carbon material, this carbon often consists of a wide variety of nitrogen species, which make almost impossible to identify which of these nitrogen functional groups are the truly active site towards the oxygen reduction reaction. And as a consequence of this fact, almost all nitrogen functional groups have been proposed as active sites towards the oxygen reduction reaction. Inspired by all these problematic, the main objective of this work consists of the selective enrichment of a pyridine activated carbon with only one nitrogen functional group with graphitic nitrogen species. As a specific objective, the preparation of these nitrogen doped carbon materials from the functionalization of activated carbon by using urea as a nitrogen precursor, the characterization of the resultant materials, and finally the testing of these nitrogen doped carbons as electrocatalysts towards the oxygen reduction reaction. Next slide shows a summary of the experimental section. As we can see here, the pyridine activated carbon has been mixed with urea in a mass ratio of 1:1 and 1:2, leading to the functionalized carbon material, which have been then heat treated at different temperatures from 700 to 1300 degrees for obtaining several heat treated carbon materials. Tables at the right show the textural properties of the resulting carbon materials, in which can be observed that these properties, this texture, porosity, is not affected, is not modified by all this procedure in comparison with the textural properties of the pyridine activated carbon, with difference below 5% or even 10%. Next slide shows the microscopy images, TEM images of the heat-treated samples, in which can be observed a disordered structure in the carbon material, which is characteristic of activated and porous carbons. Moreover, EDX mapping can be shown that an homogeneous distribution of the nitrogen and also oxygen species along the carbon structure of the pyridine material, confirming the successful functionalization of this commercial carbon. But since the most important aspect of this work consists of the selective enrichment of this material, we have followed the nitrogen oxidation state through XPS. As we can see in this slide, in the figures A and B, the bottom part, the samples obtained at 700 degrees shows three contributions. Pyridine species in blue, pyrrole species in purple, and graphitic, also called as quaternary nitrogen functional groups, as green. But when we increase the temperature of the heat treatment up to 900 degrees, we can observe a decrease of the pyridine contents while an increase of the graphitic nitrogen functional groups. And this is especially noteworthy at 1100 degrees, in which only one nitrogen species, in which only graphitic nitrogen are observed in the nitrogen spectrum. All this trend can be clearly observed in the quantification of these peaks, since as we can see in the figure C, it's possible to observe the decrease of the pyridine content and also the pyrrole content, while an increase of the graphitic content is observed by increasing the temperature of the heat treatment. Figure D, on the other hand, shows the total nitrogen content determined by the XPS. As we can see in here, the control over the carbon-urea ratio of the functionalization process enables an excellent control over the final functionalization degrees of the heat-treated sample, since the carbon-urea ratio of 1:2 always saw larger functionalization degrees than the homologous in a carbon-urea ratio of 1:1, which is especially important at 1100 degrees since the carbon-urea ratio of 1:1 doesn't show any nitrogen content, but while the carbon-urea ratio of 1:2 still show a large contribution of nitrogen content, a large contribution of graphitic nitrogen functional group. So now that we have studied the nitrogen oxidation states of our material, we can move on to the evaluation of the catalytic activity. We can see in the figure A that the optimum temperature of the heat treatment to reach the highest catalytic activity is determined to be 900 degrees, while by using a carbon-urea ratio of 1:2, the highest catalytic activity was reached after the heat treatment at 1100 degrees. Putting together all the most promising material, it can be observed that the highest catalytic activity was reached by using a carbon-urea ratio of 1:2 at 1100 degrees, the purple line. Regarding the selectivity that governs this oxygen reduction reaction, we can observe that there was no important differences regarding the carbon-urea ratio, and the most catalytic sample in figure F saw a number of transfer electrodes close to 4 during the whole potential range. So now that we have evaluated the nitrogen oxidation states and the catalytic activity of our material, we can put together this information to take some conclusions. By using a carbon-urea ratio of 1:1, the optimum temperature was reached at 900 degrees. This, after at temperatures above 900 degrees, there was a clear decrease of the catalytic activity in purple and in black after the treatment at 1100 degrees and 1300 degrees. According to the nitrogen content, this can be related to the lack of nitrogen in the surface chemistry of our material at these high temperatures. On the other hand, the highest catalytic activity by using a carbon-urea ratio of 1:2 was reached at 1100 degrees, and this is because there is still nitrogen content above 0.5 atomic percent in these samples. There are heat treatment at temperatures above 1300 degrees, there is a clear decrease of the catalytic activity from the purple line up to the black line, since the nitrogen has been completely removed after these very high temperatures. So the nitrogen content seems to be determinant in the kinetics of the catalytic activity towards the oxygen reduction reaction, but the other very important point is the type of nitrogen species that we have in our material. And interestingly, the sample that showed the best catalytic performance towards the oxygen reduction reaction was the one that only saw graphitic nitrogen functional groups in their surface chemistry, supporting and confirming the key role of the graphitic nitrogen functional groups in the catalysis of the oxygen reduction reaction. Finally, I would like to remark the most important point of the presentation. So, nitrogen-doped carbon materials has been successfully prepared from the functionalization by using urea as a nitrogen precursor in a commercial activated carbon. Moreover, the control over the carbon-urea ratio has enabled an excellent control over the final functionalization degrees of these activated carbons. The high temperature treatment leads to a selective enrichment of the carbon materials with a nitrogen content at around 0.5 atomic percent. And finally, the materials obtaining at temperature between 900 degrees and 1100 degrees show very good efficiency and catalytic activity towards the oxygen reduction reaction, approaching platinum-like performance. And finally, thank you so much for your time and for your attention. Thank you.
Hello, my name is Jacob Martin, and I'm very excited to explain some of the most recent work we've been doing on how soot forms in a flame. So why are we interested in how soot forms in a flame? Well, it's a significant pollutant. Soot actually contributes quite significantly to climate change. This is a plot of radiative forcing. It shows the usual culprits of CO2 and methane. What you might not know is that black carbon, so that's emitted from furnaces and from internal combustion engines, is also a significant contributor. Not only that, soot has been correlated directly with an increase in COVID-19 death rates, and this has been suggested to be due to damaging our lungs. So we'd really like to stop the emission of these industrial sources of pollution. But it also can be a useful material. So you can make quite useful materials out of soot. So for example, carbon black, which is a heat-treated soot, which has been heavily carbonized, is being looked at the moment for the production of hydrogen. So you're probably familiar that hydrogen is produced from natural gas with water, and steam methane reforming produces hydrogen and a lot of CO2. So people have been thinking about storing that CO2, there's some difficulties there, or using electrolysis to produce hydrogen directly from water. There's an intermediate step called turquoise hydrogen, where you use renewable energy to crack the natural gas into a solid carbon and hydrogen. And this solid carbon is carbon black and can be used in tires, in batteries, in inks, and many other things. There's also been some very interesting nanomaterials. So nanodots have been recently synthesized in flames. So by changing the fuel, different colored nanodots can be produced. And also by changing the size, they can be tuned as well. So we'd like to understand how to improve the conversion of methane into hydrogen and also how we can tune the properties of these carbon nanoparticles that are coming out. The work I'll show you today is summarized in a review that we recently had published in Progress in Energy and Combustion Science and was done in Professor Marcus Kraft's group at the University of Cambridge. So this is a schematic of how soot is formed from fuel. So the fuel, heated up by the flame, degrades into these pyrolysis products, which then cyclize into ring species and grow with acetylene being added to these large aromatic molecules. And at some stage, soot is incepted, which means the molecules cluster together and form particles. And importantly, we still do not have a predictive and general model for this process. And this is significantly problematic because we don't really know how to eliminate that soot being produced or tune the properties of new carbon nanomaterials. So in this talk, I'm going to be talking about two reactive species and why we're quite interested in them as a potential pathway to how soot forms. So the first thing is these localized pi radicals. So this work was done in collaboration with IBM Research and also with Dr. Andre Dana and Dr. Mario Camono from Italy. And in this work, molecules from the flame were extracted and then were imaged directly with non-contact atomic force microscopy. And this is really quite neat. Using a functionalized tip with carbon monoxide here being the functionalization molecule, you're able to tactile, so you're able to feel the molecules and actually see the bonding network, which is really remarkable. So this is the first molecule that I want to show you. This molecule here is an aromatic molecule, and it has two pentagons on the outside. Now, I just want to zoom in on what that looks like. And you can see that around this site, there's actually two hydrogens here and one hydrogen here. And you can see what that looks like here. This means that there's an odd number of electrons in the pi network, which means that this is a radical. Now, we know about aromatic radicals, and most aromatic radicals are this type, and these are unreactive. They do form some bonds when they're stacked, what's called a pancake bond. This is a multi-center bond, but this is not stable in the flame. Whereas we found that these localized pi radicals on these specific sites form very strong, so flame-stable bonds. We call them rim bonds because they are quite unique in that they bond around the rim, and they also allow stacking to occur. So this is a new type of aromatic bonding that we found. But I guess another question is whether these localized pi radicals are even present in the flame. So we teamed up with a group at Tsinghua University to create a kinetic mechanism that was able to explore hydrogen adding and removing from the edges. So we could actually predict how many localized pi radicals would be expected in the flame at a variety of temperatures. And we found that it is a dominant radical, so this purple line here shows it is in higher concentration in some instances than sigma radicals, where a hydrogen is removed from the edge. And it also aligns with what we see in the atomic force microscopy. So they are in a considerable fraction in flames. The second molecule I want to draw your attention to is this one here. And this is a fascinating molecule. You can see it drawn out here schematically. And when we first drew this, we thought, well, there are an even number of pi electrons, and so they should all pair up and form a closed shell species. But to our surprise, when I computed the electronic structure at a reasonable level of theory, found that the electrons like to actually to not pair up to form a diradical species, so a triplet diradical, where the two radical sites are considerably reactive. And this really demonstrates that these localized π-radicals are indeed localized to the edge because they're not able to bond through the aromatic network. And this makes them very, very reactive, these triplets. So let me explore them a little bit more. So what I mean by them being very reactive, and this I explored with Agnieszka, that these are triplets and they actually react with no barriers, and the reactivity is maintained as the molecules cross-link. And therefore, what we actually have here is a chain reaction. So we're calling this the polymerization of aromatic rim-linked hydrocarbons, and it's the first sort of kinetic mechanism that that has this kind of chain reaction, which is critical for a rapid reaction to explain soot. But we want to go further than that, and working with Lara Piscasio, we developed some very high-level calculations that were able to explore how these molecules actually collide at flame temperatures and react in the flame. So I just want to show you that video now. So you can see the two molecules are coming together. And interestingly, they are not reacting yet. They are actually interacting with dispersion forces or Van der Waals forces. And you can see them here, and there'll be a zoom up here, and you're looking for the red bond that is formed. You see it forming right there, this barrierless reaction that happens in this structure. So let me just deconstruct that a bit. What I showed you and what we found for many of these collisions is that the molecules first condense, so they actually stick together and form this dimer. And we also find that there's internal rotors, so they're able to rotate relative to one another. And this increases the chance that those reactive sites will actually find one another. And for these size molecules, there's a significant increase in the reaction rate. And we found that this is only possible for the localized π-radicals. It's not possible for a radical where hydrogen's just abstracted from the edge of the molecule. So just in summary, I showed you these new reactive sites, these localized π-radicals that allow for diradicals to be stabilized within a single molecule and allow for polymerization reactions that we think are very promising for explaining soot. So what do we do with that? Well, we can't get rid of these sites with oxygen, but we can remove them with oxygen atoms. And one way to do that could be to add ozone to the flame. And we have seen that ozone addition to engines does significantly reduce soot. Secondly, if we can remove the pentagons selectively, then we can stop soot from forming and grow graphene. And there is some evidence that we've found in microwave reactors where large graphene can grow when you get enough oxygen atoms to etch the edges. And finally, we're very interested in how we can use this new pathway to enhance the conversion of methane into hydrogen. So thank you very much for listening, and I welcome any questions.
Good afternoon, ladies and gentlemen. My name is Agnieszka Chojnacka, and I am a postdoc at Poznań University of Technology in Poland. And it is my pleasure today to show you some recent data related to dual shape pore model, which were developed to analyze the gas adsorption data of hierarchical micro-mesoporous carbons. Hierarchical porous carbons are made of interconnected micro-mesopores, which can be visible in the presented picture. And here, even the different kind of carbon precursor was used, like glucose, amine, microalgae, or soya flour. All of the samples are showing the foam-like structure, which gained a lot of attention recently due to the different field of application. These kind of materials can be used in the water treatment, water desalination, or CO2 capture, in which they can act as adsorbers, or they can be used in supercapacitor for the preparation of electrode material due to their microporosity. common method of obtaining such materials is hard templating method, in which the silica template like MCA48 or SBA15 are used. And in the first step, these silica templates are impregnated by the aqueous solution of sucrose mixed with sulfuric acid. Here the sulfuric acid plays a role of catalyst. And then such a mixture are heated to the desired temperature between the range of 800 to 1100 degrees Celsius. After that, the silica template is removed, usually by the solution of sodium hydroxide mixed with ethanol, and in this case we are able to obtain order mesopore carbons. And we adapted this synthesis to our purpose, in which we mixed the colloidal silica with glucose, after that heated it at 70 degrees Celsius, and then carbonized it at 900 degrees Celsius under the nitrogen flow. And of course, at the end, we remove the silica template with the 3 molar solution of sodium hydroxide. And in this case, we are able to obtain the template carbons labeled as STC, what means silica template carbons. For the preparation of the samples, we were using the 3 different diameters of silica. The diameter of 7, and for this set of samples we change also the mass ratio between the glucose and silica, and also for the diameter of 12 and 20. And the example silica carbon composite after pyrolysis, and also the resulting templated carbon, in which we were using the silica diameter of 12 in our mixture precursor, is shown in the right. And as you can see, the pores of spherical shape are uniformly distributed at the place left by silica template, and by the 30 measurements we were established that the average diameter of the pores is around 9.5 nanometer, which is very close to the value given by the producer. First, I would like to show you the effect of the different diameter of silica on the carbon properties. And as you can see here, for the sample in which we were using the silica template of 7, 12, and 20, all of the isotherms are characterized by the H1 type hysteresis loop, which closes perfectly at the 0.6 relative pressure. And by usage of the slit pore model, we calculated the pore size distribution, which shows the 2 main regions, the mesopore region, which is of course connected to the used silica template, and also the micropore region, which is connected to the created walls by the pyrolysis of the glucose. And what is interesting, that between the micro and mesopore regions, they are unexpected peaks, which are attributed to be artifacts because there are no noticeable feature on the isotherm that can explain their appearance. Based on the presented data, we thought that maybe our approach is not so much accurate. That's why we started to develop the new model, in which we postulated that the micropores should be analyzed by the slit pore model using the adsorption isotherm branch, while mesopores should be analyzed by cylindrical pore model using the desorption isotherm branch. So to calculate the pore size distribution, we were using the dual kernel method, in which we fit simultaneously parts of the adsorption branch, which are lower than relative pressure or 0.2, and parts of the desorption branch, which are higher than 0.2 relative pressure, using the slit kernel and cylindrical kernel respectively. Additionally, to verify our approach, we calculated the pore size distribution using the dual shape method. It is represented by the solid green curve, and compared to the pore size distribution calculated independently by individual slit pore model, which is represented by the red dashed line, and the cylindrical pore model, which is represented by the blue dashed line. And as you can see in the presented picture, we were obtain the excellent agreement between the pore size distribution calculated by dual shape model with the pore size distribution calculated by the individual models in the region of the micro and mesopores. To compare dual shape model with the slit pore model, we applied dual shape model to the samples which I showed you at the beginning. So the samples in which we are using the silica diameter of 7, 12, and 20 in the precursor mixture. And here we can see the pore size distribution results calculated from the slit pore model. This is the result represented by the dashed black line, and the results obtained from dual shape model, which are represented by the solid black line. What is interesting that the micropore regions is the same for both applied methods, because we are using the same range of adsorption range for calculate the micropores. However, for the mesopores, we found the much more narrower peaks where we are applying the dual shape method, but additionally we are able, by using of this method, eliminate totally these peaks which are labeled as artifacts. Moreover, by application of dual shape method, we can see the perfect fit to the experimental data. To compare dual shape model with slit pore model more deeply, we compare the cumulative surface area and also volume of the micro and mesopores obtained from dual shape model and slit pore model. As you can easily see, the cumulative surface area calculated by the dual model is much higher than these were calculated by the slit pore model due to the larger contribution of cylindrical mesopores. So we applied dual shape pore model to see also the effect of the glucose-silica mass ratio on the carbon properties. And in order to do it, we measured the gas adsorption-desorption for the 3 samples, in which the mass ratio between the glucose and silica was 2, 1.7 and 1.4. And as you can observe, the most evident effect of the higher amount of silica in the precursor mixture is much higher adsorption capacity of the resulting carbon, and it is very nicely visible for the calculated results. Of course, here the mesopore size is the same, because in this set of samples we are using the same template of the silica. However, all of the porous texture parameters, especially cumulative surface area and volume of mesopore, are increased almost proportionally to the content of silica particles in the precursor mixture. So we applied dual shape pore model also to the samples with surface functionalities. In this case, we took one of the samples which I showed you before and oxidized it at the temperature of 370, 390 and 410 degrees Celsius. And as you can see, the adsorption characteristic indicate that the oxidation process didn't damage the integrity of the pristine sample. However, some of the systematic change occurred, and they are visible in the calculated data. Here, as in previous case, we are using the same silica template, so the mesopore size is the same. However, all of the texture parameters are increased with the increasing of the oxidation temperature. To conclude my talk, I can say that we were successfully developed a new approach for analyzing the gas adsorption data for B-model carbons based on slit pore model and the cylindrical pore model, and we applied for different kind of carbons, differ by their density, mesopore size, and also surface oxygenated functionality. And we believe that this better representation of the B-model carbon is very important, especially in the point of view of their application. However, we didn't stop work on this model. We are still working on the develop it to be the more rigorous and more accurate, and we want to apply it for the new kind of samples in which we add additional agent for increase the microporosity. So if you are interested in this topic, please follow our updates. And I would like to acknowledge the National Science Centers for supporting presented research under the Maestro project. And I would like to acknowledge also my co-workers, especially Dr. Jacek from micro-mesoporous company. Thank you for your attention, and I will be glad to answer your questions.
Thank you very much, all the presenters. That was awesome presentations. If you wish to have a question, you could type it in the chat and let us know to whom, to which of the presenters, or you could raise your hand and I could call your name if that is easier for you. Let me get the chat. Okay. So I would like to start with a question for Dr. Karahan. You did all these studies on bacteria, but one of the things that you were, well, first of all, did you try any viruses? And excuse my lack of knowledge on biology, but how much difference that will be, and could we apply it to viruses? And also, how do you target for particular bacteria, like you said, on worms or on the body? Then you want to attack the poisonous bacteria, but not the good one.
Very good question. As far as I know, no one can target any bacteria specifically. We couldn't do either. From like last question to first, virus thing is a little more challenging. Theoretically, it should work up to an extent, especially for lipid-covered viruses, lipid enveloped ones, but not all types of viruses. First of all, there are some studies, but I never studied myself.
Okay.
Is this fair enough?
Yeah, that's good. Yeah.
Thank you.
Any more questions? Then I carry on with another question myself. From to Dr. Quilez-Bermejo. With the nitrogen, or you were doing nitrogen on activated carbon, but a lot of the carbon nanotube community have been using a lot of nitrogen, and they get these different structures when they introduce nitrogen. The layers inside break and they call bamboos. So there is a lot of research on putting nitrogen in carbon nanotubes. Is there a chance of using that as well as activated carbon, as you could get, is there any potential to do that?
Thank you for the question. Yeah, it's the textural properties, the structure of the carbon material is almost as important as the type of nitrogen that you have in your material. But to my mind, if you don't have active site, the structure or everything else is not as important. You need some nitrogen or some heteroatom defects or something that can disturb the electronic structure of the carbon material in order to make the chemisation of the oxygen molecules easier. But for example, the carbon nanotubes has this different hybridization of the carbon atoms, which can also increase the catalytic activity.
So have people tried?
Yeah, there are people that have tried to do nitrogen doping on carbon nanotubes, yes, and the catalytic results are quite good.
We have a question for Jacob. Have you or someone else done any studies of the energetics of the competing reactions surrounding breaking the soot formation, like the pentagon?
Yeah, so we've actually published a series of various reactions between aromatic molecules So we do know how different aromatic radicals will break down at flame temperatures. We've computed the rates as a function of temperature for those. We have looked at, there's been some preliminary work on some of the reactions that would break some of those bonds that form soot. And I think the most promising ones are the ones where oxygen atoms are used to basically remove the carbon atom at a pentagon site. But I've also seen recently in a conference someone showing that CH can actually insert and turn a pentagon into a 6-membered ring, which could be another potential route. So that's with a CH radical.
Okay.
May I ask a question here? It might be easier to speak up.
Go for it.
Yeah, along those lines, very curious. No, very nice presentation. So one quick question, since you brought up this question for Jacob. I'm just curious. It's this selective elimination, as I understood it from your presentation, the possibility of eliminating selectively these defects as they form by adding oxygen at the right time. It has, you mentioned this, but I was wondering what's the effect of temperature and pressure on those. Again, I may be speaking of ignorance in your model. I apologize for that. But have you looked at when these radicals are formed and when they can be, these defects can be eliminated as a function of temperature, pressure, or confinement? That would be my second question. As these things are formed, you're forming them in, as I understand, in free space. You don't have any confinement effects. But if you were to form soot or in some porosity or some confined form, are we at the stage, or could we lead the research towards understanding the formation, what forms, and whether we can selectively control that formation? Is it making sense? Or?
Yes, no, that's some really good questions and also interesting ideas. So in terms of temperature and pressure, we find that in terms of pressure, it's very interesting. It appears as if it's second order in the aromatic molecule concentration. So this is what led people to think that it's a physical condensation, because basically it's second order in the monomer concentration. And we think that that's due to this physically enhanced soot process. So the molecules sticking together and then reacting. And so pressure, increases soot formation second order in aromatic concentration. So it really increases soot a lot. In terms of temperature, there's a bell curve. So there's a temperature at which it's maximum, around about 1,800 Kelvin, I suppose. And then higher temperatures, it goes away, and lower temperatures, it also stops forming. So below about 1,450 Kelvin, soot won't form. And so there are ways of doing low temperature combustion and also very, very high temperature to stop soot from forming. I think that idea of confinement is particularly interesting, and I haven't seen a lot of work done on it. It does remind me of a paper I read where they actually made little channels down to a platinum catalyst, and they showed that the carbon wasn't deposited on it because there were these very small channels that stopped the condensation and the formation of coke. So there's definitely some potential there. And an interesting suggestion. Thank you.
All right, thank you.
Any other questions?
Well, there's time. I do have another one.
Yes, go for it, Carlos.
Yeah, maybe again, very interesting work on this one. There's literature that talks about this, but in your experience from your models, these defective sites, 5-member rings, and I presume I looked at your images, 7-member rings, 4 to 8 rings. They have some thermal stability. But if we were to look at temperature effects only, at what temperature do the transition from actually the elimination of these defective sites, if you will, occurs? Has that been
right? Yes.
calculated?
So I'm assuming you mean that basically there are kind of reactions at the edge, such as kind of Stonewall's transformations, almost, where basically you get the removal of a pentagon, and actually two pentagons can combine at an edge and form a hexagon. And so we've done some work where we actually look at the growth versus the rearrangement around the edge using a kinetic Monte Carlo model. And we also looked at heptagon integration as well. And we found that, that the integration of pentagons is faster than their removal. And this means that you get a couple of pentagons that are persistent. Once they're actually completely embedded with hexagons, then they are not able to be removed with any kind of rearrangement reactions. But there potentially is something to say about maybe tuning the those sort of edge reactions and trying to anneal out pentagons. And I think that's what happens in these microwave reactors with graphene formation.
By perturbing when you have the microwave?
I'm not entirely sure what happens. I think it's just a longer residence time. That's what I'm thinking at the moment. It just, you know, there's just longer time to anneal things out.
Okay, okay. And if I may, I mean the plug thing, that also you mentioned there's some hydrocarbons that could also lead to this, I guess, attachment to the edges and reduction of the defect formation, if we want to call them defects. Can you comment on that? What hydrocarbons have been investigated?
So methyl radicals, so CH3, has been shown to embed, as well as the methylene, so the CH radical. Those two have been shown to add to these types of sites and change them from a pentagon to a hexagon. I can send you the paper for that.
Yeah, I would appreciate that. We appreciate learning that more. Your work, really very interesting work. Thank you very much. All of them are very thought-provoking, but thank you. I think I captured enough of the time. I appreciate your answers.
Well, we can continue in Gathertown, and the presenters will be there. We can continue asking questions. They were all excellent talks with a lot of things to think about, and a lot of new ideas and interesting work. So we have 2 minutes left. I will start wrapping this up. I would like to thank you all, the presenters, and congratulate you for all your prizes. They were excellent talks. Thank you everyone for attending this webinar. We have one more carbon webinar before the end of the year. That will be on Monday, the 13th of December, or Tuesday, the 14th of December, depending where you are in the world. And it will be given by Professor Yasuhiro Yamada. We will also announce the winners of the poster prizes from our previous webinar. Thank you to all the judges who took part of it. I would like to, Jake will be put the Gathertown post in a minute, but I would also like to let you know that the Carbon 2022, which will occur in London next year, have opened for early abstracts. So just go to carbon2022.org and there will be some information there. But now, if you are still inclined to continue the discussions and have more questions that you wanted to ask, we will all gather in Gathertown. The link is now on the chat. So if you can just click there, please put your name so that people can talk to you and know who you are. And thank you everybody for this awesome seminar.